Hello everyone, my name is Poirazi and I'm a research director at Forth in the island of Crete in Greece. The purpose of this talk is to summarize what you have already learned about real neurons and ways to model them. I will also briefly discuss compartmental modeling of dendrites, which we haven't been taught about today. And the material might be complex at some points, but you shouldn't worry about that because the goal is to give you a perspective on why dendrites matter. So let's get started. As Yubi told you today, the brain is complicated. Processing takes place at many levels, from the molecules to the dendrites, the circuits, and all the way up to behavior. And in order to understand the brain, we must understand how information flows at all of these levels. Today, however, you mostly heard about the microscopic level, namely how to model synapses and individual neurons. But there are also different levels of modeling, starting from the microscopic to the mesoscopic and the macroscopic, with different types of models. Today we focus primarily on single neuron modeling. If we take a short outlook on the history of single neuron function, we will start with models that date back to the 1940s. At the time, neurons were thought as linear thresholding devices, where electrical current arriving from other neurons is weighted according to different synaptic strengths, and if the sum surpasses a given threshold, then an action potential is generated. It wasn't until the 1960s that Wilfried Ruhl, with his beautiful modeling studies, showed that the passive properties of dendrites allow these structures to compute in nonlinear ways. After the discovery of active dendritic properties in the 1990s, we now know that dendrites can compute highly nonlinear functions within compartmentals in a compartmentalized way within dendrites, thus empowering single neurons with added computational power. So let's take a look at these different approaches. We start with the artificial intelligence neurons, as you termed it. This model was introduced in 1957 and it was termed a perceptron. It operates on a discrete time scale with a tick equal to the refractory period of the neuron. The input is the activity of different neurons, which is weighted at synapses, summed at the cell body, and passed through a thresholding nonlinearity at the axon. The output is binary, 0 or 1, and each connection synapse has an attached weight which can be plastic. Both positive and negative weights can be incorporated in these models, representing excitatory and inhibitory synapses. So what are these mathematical models representing neurons good for? Well, perceptrons have been extensively used in the machine learning field, primarily to solve classification problems. They are very good at finding a hyperplane to separate two different classes of examples, for example, apples versus oranges. More importantly, they served as the foundation for the birth of artificial neural networks. Many of you have also heard them being termed deep neural networks in cases where the number of hidden layers is high. These artificial neural networks can be trained to solve many complex tasks without obeying any task-specific rules. They are excellent global approximators and have been already shown to achieve performance that surpasses that of humans in many challenging tasks. So what are these artificial neural networks good for? As we said already, they're extensively used in machine learning to solve challenging tasks, and they can offer surpass human performance, for example, the AlphaGo and many gaming applications. They're also important in inspiring research approaches in neuroscience. By studying the structural and the learning rules that work best in these systems, one can think of ways that the brain may also be using to solve similar problems. Negative aspects include the high complexity of some of these deep neural networks, where the number of trainable parameters is often too high to infer any meaningful insights, similar to real brains. And also, these models are not very good when one is interested in understanding single neuron dynamics, and especially those that happen at the subthreshold level or within compartments such as dendrites. So how can we improve these models? Well, obviously by adding details. And you already heard about circuit models of neurons. These models are based on an analogy between elements of the neuronal membrane and electrical circuits. You already heard, for example, that the membrane can be simulated as a capacitor, Different channels can be simulated as resistors because current flows through with some resistance. The pumps that maintain the gradients of ionic concentrations can be simulated as batteries. And we also have voltage-gated ion channels whose conductance is dependent on the voltage of the membrane, and those can be described as adjustable resistors. And Huxley and Hodgkin were one of the very first people that described the dynamics of these adjustable resistors with ordinary differential equations. So with these tools, mathematical tools at hand, we can now describe neurons as elaborate RC circuits, where each of the conductances and the mechanisms, synaptic mechanisms that are found in neurons are described with these electrical elements. Obviously, these models can be highly complex, requiring many different parameters to describe the dynamics of these membrane mechanisms. But as you already heard from Richard, you can reduce these models to account only for those aspects that are most critical. For example, the Hodgkin and Huxley equations describing sodium and potassium conductances that underlie action potential generation can be replaced by a threshold for spike emission that is followed by a reset to a fixed potential. Other mechanisms, other conductances, that is, for example, after hyperpolarization or H current, can be completely ignored or approximated with linear dynamics. And as you already heard, we can devise a number of variants of these simplified models, which we can account at will for different properties, such as, for example, adaptation or different time scales. So what are these reduced models good for? I'm just highlighting one example provided by Richard. The main idea, the main property that is captured by these reduced models is what happens at the cell body of neurons in response, for example, to current injection. These models are very good at capturing the dynamics of somatic activity. And this was evidenced already by comparing the accuracy of these models in both pyramidal and fast spiking interneurons, which can be as high or even higher compared to detailed Hodgkin and Huxley models that incorporate a much higher number of parameters. But as we said, these models are limited to somatic responses. They can predict somatic output with very high accuracy. They can also be used to predict sample dynamics even under noisy conditions, assuming that the input is limited to current at the cell body. And they can also be used to approximate synaptic dynamics and plasticity as described by Richard through the modeling of, for example, STDP, assuming that synapses are happening at the cell body. However, neurons are morphologically complex. They come in great variety in their elaborated dendritic trees. They are also highly complex in the type of computations that can be supported by dendrites. For example, they have the ability to generate different types of regenerative events, also termed dendritic spikes. Calcium spikes, they appear mostly in the apical trunk and they are highly slow in terms of their kinetics. Sodium spikes, they are fast rising and they look like action potentials and can be found throughout the dendritic tree. NMDA plateau potentials, typically found within thin dendrites, are slower and they have these very slow waveforms that are distinctive of NMDA activity. All of these events are typically ignored in reduced models simply because we don't account for dendrites at all. Why should we care about these dendritic events? Are they important and should they be incorporated in the reduced models? Well, my answer would be yes. And there is strong evidence coming from experimental studies as to why this is the case. In a beautiful experiment performed in the lab of Matthew Larcum in Humboldt University, they asked mice to detect the presence of a given pole based on their touch, based on whether the whiskers could touch this pole. So they observed, while imaging in the barrel cortex, that when the animal correctly reported the presence of the pole, there was a strong calcium activity in the distal dendrites of cortical neurons. When the animal failed to detect the presence of the pole, this activity was not seen. So they hypothesized that maybe this dendritic activity is important for sensory perception so that the animal was able to understand that the pole was present. To check this hypothesis, they applied inhibition in the distal dendrites of cortical neurons and observed that the ability of the animal to detect the pole reduced substantially. They also did the opposite experiment, where they increased the excitability of the distal trunk through optogenetic stimulation and noticed a significant increase in the ability of the animal for sensory perception, namely to detect the presence of the pollen. So these experiments highlight that the dendritic spikes in the distal tough dendrites of layer 5 pyramidal neurons are causally linked to sensory perception. Therefore, the dendritic spikes are important and they should be taken into account in models of single neurons and circuits. Moreover, there is a very nice review summarizing the compartmentalization of the dendritic processing. Dendrites, for example, compartmentalize electrical integration, the release of neurotransmitter, chemical integration, local plasticity, and protein synthesis. This compartmentalization enables higher processing and storage capacity, and as a result, single dendrites have been suggested to act as fundamental functional units in the brain. Again, all of these features are typically ignored by reduced models. So what can we do about this? How can we account about the dendritic nonlinearities, the dendritic processing in computational models? Well, there is an entire world that goes beyond leaky, integrate, and fire that takes into account the dendritic modeling. How do we do this? We need to add even more details, at least initially. So we start with a detailed morphological reconstruction of a neuron. We translate this morphological reconstruction into compartmental models, and we describe the electrical activity of these models using cable theory and circuit theory as done with reduced modeling. In this case, each part of the membrane is described as a circuit, an electrical circuit, and we have many of those connected together to provide a more detailed description of the electrical activity throughout the entire dendritic tree. So what are these dendritic models good for? I just want to give you a few examples with the purpose of highlighting the power of dendrites and not necessarily to explain exactly how we model them. If you're interested, there is a lot of literature out there. So I'm starting with a model that I developed when I was a postdoctoral fellow with Bartlett Mell. In this model, what we did was to take reconstructed morphology of a CA1 pyramidal neuron and incorporate as many ionic conductances as we knew were present in these cells, as well as synaptic receptors. We validated this model against a lot of experimental data, and the question we were interested in is how do dendrites integrate their inputs? Remember that at the time, back in the early 2000s, dendrites were thought as passive devices, and we believed they had much more power than that. So to answer this question, we delivered synaptic stimulation within two separate dendrites or within the same dendritic branch, the same exact synaptic stimulation, and asked how do these two scenarios result in responses at the cell body. What we found was that if we separate the two stimuli within different dendrites, then what the cell body saw was a linear summation of these two synaptic inputs. When these inputs were placed within the same dendrite, then the response at the cell body followed a sigmoidal activation function, which meant that if you place the same input within the dendrite, it's integrated in a different manner than if separated between different dendrites. The summary from these studies was that location mattered. Synaptic inputs within a branch are nonlinearly integrated, and this nonlinear integration is missed when ignoring dendrites. In fact, we significantly underestimate the computational power of single neurons if we don't consider these nonlinearities. This prediction came out from a compartmental model and drove the interest of the scientific community, and soon afterwards, two experimental studies verified that indeed synaptic integration within cortical dendrites, both in the basal dendrites and the apical dendrites, follows a sigmoidal activation curve. And this is an interesting, or in my opinion, an important contribution of compartmental models, which is to drive the interest of the scientific community so that then we further our understanding with respect to how dendrites compute their incoming signals. Similarly, Alexander Zilivaki, a talented student in the lab, asked the same question for interneurons. We all know well that interneurons are typically treated as point neurons, namely we believe that they simply sum their incoming signals at the cell body and produce an action potential. And in fact, Richard already showed that reduced models can approximate interneuron firing very well. But Alexandra thought otherwise, so she developed a detailed compartmental model of fast-spiking basket cells in two brain areas, the hippocampus and the prefrontal cortex, for 8 different morphologies, and she repeated the experiment that we did in pyramidal neurons, placed synaptic inputs within different dendrites, and asked, how do these interneurons integrate their signals? To our surprise, he found 2 types of dendrites. One group of dendrites was following a sigmoidal integration, much like pyramidal neurons, because they could support the generation of dendritic spikes in their dendrites, and other dendrites summated inputs sublinearly, because they couldn't generate dendritic spikes. These 2 types of integration in interneurons were found in all of the 8 morphologies that we studied. These findings, these predictions, were soon verified experimentally, in fact, a few months later, by showing in PV cells of the hippocampus a similar nonlinear integration. So in these cells, which are not necessarily fast-spiking basket cells, but they are PV positive cells, the experimentalists found that there are 2 types of responses. One that is supralinear, similar to ours, and another one that is mostly linear. So these modeling and experimental studies highlight that even the dendrites of interneurons can perform nonlinear computations, and therefore maybe treating these cells as point neurons would be a wrong assumption to make. Another interesting prediction that came from compartmental modeling is that of neurons, pyramidal neurons and interneurons acting as multi-layer, in fact 2-layer artificial neural networks. So what we showed in pyramidal neurons is that a detailed compartmental CO1 pyramidal cell can be described by a 2-layer artificial neural network where we design dendrites to have a sigmoidal nonlinearity similar to that of the cell body, and training this artificial neural network to predict the firing responses of detailed compartmental neurons. We then tested whether an artificial neural network can capture the firing rate of the detailed compartmental model with high accuracy, and found that indeed this is the case. We can describe the firing rate in response to many different synaptic stimuli very well with a 2-stage artificial neural network. Similarly, Alexandra showed that in interneurons, an artificial neural network with 2 types of nonlinearities, one that is sigmoidal to describe the spiking dendrites and one that is aprilar to describe the rest of the dendrites, is a much better description of the activity of a fast-spiking basket cell. In other words, these nonlinear models can capture the firing rate of these fast-spiking interneurons with higher accuracy than a respective point neuron model. It should be highlighted, however, that in some cases, the linear point neuron models are as good or pretty good compared to the nonlinear ones. And therefore, this is not to say that you should necessarily go with a detailed description of a model. It all depends on how accurate a description you're looking for either an interneuron or a pyramidal cell. Sometimes a linear approximation might be good enough. Importantly, what I wanted to highlight today is what are the functional implications of these 2-layer artificial neural networks. In a study that dates back to the 2001, we measured the capacity of linear versus nonlinear models in order to have a better understanding of why would it be important to account for dendrites. And what we found is that you have an increase in the storage capacity of these neurons, also if you put them into circuits, which is at least of 1 order of magnitude. So incorporating dendrites in models is important because it would increase the storage capacity of your model and similar to the neural tissue by at least 1 order of magnitude. What is another interesting property that one can learn by accounting for dendrites? This came from recent studies in human neurons, where we use these types of models to discover new types of computations that would have been entirely missed if we didn't account for dendrites. In a beautiful study that was performed in the lab of Matthew Larcombe, Albert Guidon, a talented postdoc, looked at how dendrites of layer 2-3 pyramidal neurons in human slices integrate their signals. So he applied synaptic stimulation in the dendrites of these cells and recorded their responses. And he found a new type of dendritic spikes, which is shown here and is called a D-cup, because it's primarily dependent on calcium. What is interesting about this dendritic spike is that as you increase the stimulation intensity delivered to the apical branch, then the amplitude of these spikes becomes smaller and smaller. This is something that has not so far been seen in other type of pyramidal neurons. Now, what does it mean that the amplitude becomes smaller? It means that the integration that takes place in this branch is not the typical sigmoidal one, but rather a different shape. The shape of this nonlinearity is described here by the impulse function shown here, where weak stimuli do not induce any dendritic spiking. At the ideal stimulation strength, you have the maximum amplitude of the spikes, and as the stimulus strength increases, the amplitude also drops. So this interesting transfer function that was discovered in human neurons did not have a purpose. We didn't know what could be the type of computations that are solved by this function. And we thought that these properties are ideal for solving a type of computation that has thus far been attributed to neuronal networks. What is this computation? The XOR function. The XOR function essentially says that if you have two pathways coming to a gate, if none of them are active, then the gate remains closed. If any of them is active, then the gate opens. But if both of them are active, then the gate is closed again. And this type of nonlinearity seems ideal for solving this problem. Now, to answer this, to test this hypothesis, a talented researcher in the lab, Nasi Paputi, developed a detailed biophysical model of layer 2-3 pyramidal neurons and simulated this type of attenuation in the amplitude of the dendritic spikes. Because we don't know the exact kinetics or mechanisms behind the spikes, we just simulated this decrease phenomenologically. We then stimulated two different pathways delivered to the apical dendrite of this neuron and asked whether this dendritic computation can solve the XOR problem. So what we found is that if you stimulate the first pathway, you have a strong response both at the dendrite and at the cell body. But if you stimulate both pathways together, then the response of both the dendrite and the soma decreases substantially. Therefore, this property of the dendritic spikes can indeed be used to solve the XOR problem. Even more interestingly, if you apply inhibition onto this branch, you can further increase the computations that can be implemented by these branches. For example, if you deliver inhibition within the branch, then you reduce the net excitation, therefore returning the activity within this branch to a higher level, and also inducing a stronger response at the cell body. If inhibition is placed closer to the cell body, this response is slightly different, essentially showing that a neuron can explore all of these different mechanisms to expand its processing power. The conclusion from this study is that a human neuron, and in fact its apical dendrite, can solve the XOR problem, a computation that was thus far believed to be solvable only by artificial neural networks of at least one hidden layer. Now, the question is, do we need to go all the way to build such complex compartmental models to account for the properties of dendrites? And the answer is not. There are also ways for reducing this complexity and building more simplified models of dendrites. And I'd like to highlight just a couple of examples from our lab. George Kastelakis, a talented postdoctoral fellow, developed a circuit model of the hippocampus. Let's say it's an associative area network model where he used integrate and fire models and added a couple of dendrites to them. I don't have the time to go into details, but the idea is that he used the sigmoidal activation transfer functions that we described in our detailed compartmental models and added these impulse functions to the integrate and fire neurons. He did this for pyramidal neurons, and also at later models, we also incorporated nonlinearities in interneurons. And we use these models to ask different types of questions that take place at the circuit level. These models, because of their simplicity, can also account for plasticity rules, as Richard already highlighted in his talk. The communication between neurons happens at synapses, and synapses are plastic. We already learned from Richard about the different factors that contribute to synaptic plasticity. For example, it could be the relative timing between backpropagating action potentials and local synaptic inputs. It could be the presence of different neuromodulatory agents, or it could also be the calcium concentration that is found locally near a synapse. This is just a short summary of the different types of plasticity rules found in synapses. In these models, in the model developed by George, we incorporated different types of plasticity rules, including multi-scale ones. I don't have the time to go into details again, but I just wanted to highlight that there are many different types of plasticity that can be added in this model. For example, local LTB and LTD that can be dependent on the availability of proteins. Homeostatic plasticity, which scales the weights up or down depending on the net excitability of the circuit. Plasticity of neuronal excitability that takes place at the cell body. The cell bodies of neurons that learn a given memory, we know that they remain highly excitable for several hours. And we can also incorporate axonal rewiring, namely the ability of presynaptic axons to test potential postsynaptic partners, form synapses there, and retract them if they're not strengthened within a given timeline. So in these models, we can incorporate all these types of plasticity rules and then use them to ask interesting questions. One interesting question that we wanted to ask is, how do memories interact across time? For example, think of recalling what you did today. If you try to remember, for example, where you parked, you will notice that the events that happened just before or right after you parking your car will also be recalled. How is this interaction between events that take place close in time happens at the cellular level? This is what we were interested in finding out. So we designed our model so that it learned two distinct memory events and then looked at the population of neurons that capture these memories. This population of neurons is typically termed the memory engram. And we did that when spreading the different events across different time intervals. Now, what our model predicted, that is, if you look at, for example, events that are separated by 5 hours, then the neurons that store these events, they have high overlaps, neuronal overlaps, which means that they're stored in common neurons. And not only these neurons are common, but the dendrites within which the axons that carry information about these two memories are common. Namely, these axons form clusters within the same dendrites at both the 5 hours intervals and even at shorter time intervals. And this prediction of the model is in line with experimental data that came out about the same time from the lab of Alcino Silva, where they also showed that if you look at memories separated by 5 hours, then a similar overlap at the neuronal population is seen to that of the model. So our model essentially predicts that the mechanism by which two memories become linked across time, such as in episodic memories, are both at the neuronal and the dendritic level, and they amount to overlaps. Overlaps, namely being stored in the same neurons, and dendritic overlaps, namely being stored in the same dendrites. Another interesting prediction that came out from this model has to do with resource savings. As we said, we trained our models to form memory engrams. And in this place, instead of looking at how these engrams interact, we look at their properties when adding interneurons with nonlinear dendrites. What are the properties of these memory engrams? For example, the size. How many neurons capture a given memory when these interneurons have linear dendrites or nonlinear dendrites? So we found out that you need fewer neurons to capture exactly the same information if fast-spiking basket cells come with nonlinear dendrites. These fewer neurons also fire at lower firing rates. So nonlinear dendrites make memory engrams fire with fewer spikes. And the sparsity of these memory engrams is substantially higher when we incorporate nonlinear dendrites. All of these properties amount to resource savings. Namely, the brain, or in this case the model, predicts that a given memory can be learned with fewer resources when we take into account the dendritic nonlinearities in interneurons. So to summarize, I hope I have provided you enough examples to give you a perspective on the advantages that can be provided by accounting for dendrites in individual neuron models. Dendrites allow individual neurons to process signals locally in independent nonlinear ways. They can support plasticity rules that are different from those at the cell body, and they can provide multiple types of nonlinearities that can be utilized to solve different types of problems, for example, the XOR problem that we highlighted in human neurons. At the circuit level, dendrites can subserve the linking of information across time, and they can also allow the storage of memories with fewer resources. All of these dendritic features that are shown to provide benefits in models of neurons are also likely to provide benefits to machine learning systems and artificial intelligence systems, and are therefore probably useful to consider. Now, in summary, what we discussed today was a brief overview of artificial neurons, perceptrons, and their pluses and minuses. These are good for machine learning, but they cannot offer much insight onto single neuron mechanism. They can be used to provide some design principles and learning rules for inspiring future experiments. We also discussed about reduced single neuron models by Richard, which are great for addressing questions related to somatic activity in the absence of dendritic nonlinearities. They are of low complexity and can therefore be used in large-scale networks, but cannot account for localized dendritic effects or localized plasticity. I've also given you a few examples of compartmental models, which are great for addressing questions related to subcellular computations, like dendritic integration, dendritic plasticity, and they can also provide readily testable predictions because they account for the complexity of the real system, but do not account for detailed molecular cascades, and a major drawback is that they are computationally expensive. At the end, we also saw a few models, reduced models with dendrites that can replace to a large extent the detailed compartmental ones, and they can take into account the dendritic nonlinearities and localized plasticity rules. These models are of medium complexity and therefore can be used in large but not too large scale circuits. And I would like to end with a conclusion that comes from a recent review article on dendrites, but I think applies for modeling in general. And the conclusion is that there is no single answer to the quest of finding the best model. This quest depends heavily on the question or the questions being asked. If we can give a universal advice, that would be based on Occam's razor. The best model is the one that is detailed enough to address the question of interest while requiring the smaller possible number of parameters. If you're interested more about dendrites, then you can check out our recent review on that. So with that, I'd like to thank you very much for your attention, thank the member of my lab that worked on all of the modeling aspects that I presented today, our collaborators around the world, and our funders. I hope this was an interesting lecture for you. Thank you very much.